So we're going over my cell biology in a nutshell, life science notes. So we were learning about the cell membrane, the phospholipid bilayer of cell membranes, and these consist of a bilayer with two phospholipid molecules facing one another with a polar head and a nonpolar tail of fatty acids. And we were learning that inside this bilayer of the within the in between these molecules, you have interactions with proteins, and the proteins, if they're integral, they're within the bilayer. So the polar sections of the molecules of protein interact with the polar parts of the phospholipid molecule, while the nonpolar parts of the protein interact with the nonpolar or the hydrophobic parts of the phospholipid molecule, more towards the interior of the bilayer. So in the hydrophilic parts of the molecules are towards the exterior of the cell or towards the cytoplasm of the cell, but on the external part of the molecule, the bilayer. So again, we have it says the membrane spanning portions of integral proteins consist of 20 to 25 nonpolar amino acids, which are alpha helical in shape. So they're transmembrane proteins. Some transmembrane proteins span membrane more than one time, but at multiple regions. So depending on what kind of protein, how big it is, what it's for. Most transmembrane proteins have been modified by carbohydrates binding, also exposed on the surface of the cell, participating in cell-cell contact and communication. Proteins can be anchored to the cytosolic face of the membrane by addition of 14-carbon fatty acid, myristic acid, to the amino terminus, or addition of 16-carbon fatty acid, palmitic acid, or a 15 or 20 carbon prenyl group to the side chains of cysteine residues. So alternatively, proteins are anchored to the extracellular face of the plasma membrane by the addition of glycolipids to their carboxy terminus. So glycolipids are carbohydrates binding to lipids. So I guess via their carboxy terminus. So the ends of the molecules interact. So you have extracellular interactions and cytosolic or intracellular interactions between proteins and the bilayer components. Small uncharged molecules diffuse freely through the phospholipid bilayer. Small uncharged molecules, but the membrane is impermeable to large molecules, which are polar, especially, and these are like glucose and amino acids, as well as ions. So these protein transport channels within the membrane help carry across these larger polar molecules as well as ions via ion channels. So here shown you have glycerol, ethanol, water molecule, carbon dioxide, oxygen, and nitrogen. N2. So says these are easy molecules to pass through the bilayer. So I mean, they're relatively easy and they don't require any kind of a channel. They could just freely roam through the bilayer from the outside to the inside, vice versa, of the cell. Large polar molecules and ions such as calcium, sodium, hydrogen, amino acids, chlorine, glucose, they require some sort of channel to get across the layer, and therefore energy. So O2, CO2, which are considered nonpolar since they're relatively symmetrical, and they're soluble in the lipid bilayer and cross the membrane. Uncharged polar molecules like H2O also, and the large polar molecules do not pass. Ions as well, unable to diffuse through the phospholipid bilayer, regardless of size, because they're charged. Even hydrogen plus ions do not pass freely through the bilayer. Channel proteins form open pores through the membrane, allowing free passage of molecules of appropriate size. Ion channels allow passage of inorganic ions such as sodium, potassium, calcium, chlorine across plasma membrane. Once open, channel proteins form small pores through which ions of appropriate size and charge cross the membrane by free diffusion. The pores formed by these channel proteins are not permanently open, rather, they selectively open and close in response to extracellular signals, allowing cells to control movement of ions across membrane. Such regulated ion channels have been particularly well studied in nerve and muscle cells, where they mediate transmission of electrochemical signals. In contrast, channel proteins, carrier proteins selectively bind and transport specific molecules such as glucose. So rather than opening up channels, carrier proteins act like enzymes facilitating passage of specific molecules across membranes. Carrier proteins bind specific molecules undergoing conformational change, opening channels, and then the molecule is transported across the membrane and released on the opposite side. Channel proteins form open pores through which molecules of size appropriate ions move across the membrane. That's in A diagram. B diagram, carrier proteins selectively bind small molecules or any molecules that are transported undergoing conformational change of the transport protein, releasing the molecule on the other side of the membrane. Molecules transported in an energetically unfavorable direction across the membrane or across a gradient against a gradient of concentration, so lower to higher concentration, is coupled to ATP hydrolysis, so as a source of energy. A process called active transport. Carrier proteins provide a mechanism through which energy change associated with transporting molecules across membrane is coupled to use or produce other forms of metabolic energy, such as enzymatic reactions, coupled to hydrolysis or synthesis of ATP. Free energy stored as ATP is used to control internal composition of the cell, as well as drive biosynthesis of cell constituents. ATP changes to ADP, a phosphate group is released, and hydrogen goes from low concentration to high concentration. Here, same thing. Let's say an example of active transport. Energy derived from hydrolysis of ATP is used to transport hydrogen across the gradient, binding hydrogen accompanied by phosphorylation of the carrier protein and inducing a change of conformation, driving hydrogen transport, releasing hydrogen, and hydrolysis of the bound phosphate group restores the carrier to its original conformation. So hydrolysis is a water added